record. Now I can share my screen. So on Pearson, if we go to our course, I kept most of the things where they were, but I did move one thing. So here's the calendar, right? So I did move your lymphatic system homework was due, or lymphatic system lab was due yesterday. I did move the quiz till tonight, so that anybody who wanted to wait until after today's lecture to take the quiz can do so. Now you also have to realize that the respiratory system homework is due tomorrow, because we're going to start the respiratory system today and finish it tomorrow. And the respiratory system quiz is due Sunday. Right, so that stuff is coming up, as well as the exam is due May 3rd on Blackboard, or I'm sorry, on Moodle. So I did give you guys extra time to do the exam, figuring that you might want to listen to some of the lecture before you do the exam. Are there questions before we get started today?
The new syllabus that's posted, it says are we doing the fetal pig dissection next week?
Yes. So
Okay.
so this is the one you're talking about, right, Melissa?
Mm-hmm.
Yeah. So what we'll do today, we will finish up the lymphatic system, start the respiratory system, finish the respiratory system tomorrow. And what I'll do is I'll go over the lab 12, which there's a PDF that I posted this morning. So I'll go over that with you guys. So it'll go through how to do the actual lab and what to do. And there are a few charts on there that you need to fill in and hand in as part of the lab. And then next week, we'll do the fetal pig dissection. I'm wondering if we should do that at like 2 o'clock, like do an hour of lecture on the digestive system so that we know where some of those organs are, because a lot of what you'll see on the pig will be the digestive system. And then maybe at 2 o'clock, start the dissection. Because I know on Thursdays, some people can't make it on Thursdays. So I'd rather do that on Wednesday when I have a bigger group of you here. So again, to recap again, today we will finish the lymphatic system and start the respiratory system. Tomorrow we will finish the respiratory system and discuss the respiratory lab. Then next Wednesday, May 5th, we will start the digestive system at 1 o'clock. And then at 2 o'clock, we will dissect our pig.
That makes sense.
Does that sound
Thank
good?
you. Yeah, thank you.
I have a question about the lab that we
Sure.
just did. Well, that's due tomorrow now, I guess. I think you
Okay.
just said. At the top of it, it said something about clear your workspace, and I wasn't really sure what that meant. It was something, I think it was trying to like refer me to be looking at something before taking like the test, basically. And I couldn't figure out how to access it. Did anybody else see that? I don't know if it was like cadaver pictures, because once I got onto those sections, I was like, okay, what is this? You know, what exactly are they looking for?
Yeah, I see it too.
Yeah, I was confused. In some of the pictures, I was like, I don't know what body part that is. So
No,
I guessed a
exactly.
few things.
Yeah, me
And then my
too.
final
Yeah, I
guess
didn't
was
do
just
good.
a
I was
no
so glad we
idea.
had 2 chances.
I did not
Yeah.
do well on that one.
Not the first time around, no. But it was
I had
like,
a problem.
yeah. One
I
was
had
like
a problem with
lymphatic-
2.
Oh, go ahead, sorry.
Sorry. There's 2 on there that they won't take the right answer, and eventually it took it just put a limp. That's all.
Hmm.
And
Yeah,
it's just
same.
so strange. I
Or
took a
just
picture of them.
lymph
Oh,
node.
yeah.
Yeah.
I remember that one.
Yeah. One of them is
What was
the
passing through. Yeah.
brachial lymph nodes. It won't take the whole word, so I kept getting it wrong, and I know it's the right thing, so I just put lymph, and it took it. And then the other
Yeah.
one, The
Yeah, there
other
was a,
one is inguinal. The inguinal lymph nodes,
Mhm.
same. You just put lymph and take the right answer.
Or the one
Yeah,
that was the
that.
abdominal cavity, it was just lymph node. That's all that they wanted. And I was really trying to be specific. But anyway, I was glad that we had 2 chances at that one.
Okay, well, that's good feedback. I need to look closer at that over the next day and see exactly, so I can see exactly what you're talking about. But I certainly will take those things into consideration when I'm posting your grades. Um,
I mean,
if
so
that
there was
wasn't the
something that was
best
written
representation.
up, oh, sorry, up top though, about, like I said, like referencing something before we started or something. Do you guys know what I'm talking about? I can't remember exactly how it was
Yep.
worded, but
I
and
just
wasn't
kind of
sure
went
how to
along
access
anyway.
it. Okay.
I remember seeing that. I thought it was just a technicality, so I didn't really pay
Okay.
much attention.
Same here. I didn't really think it had much to do with it.
All right.
Okay.
And it wasn't until I hit the, cadaver photos that I was like, oh my gosh, maybe that's what it was referring to. And, I didn't know how to get there to reference these things before answering these questions. So I'm glad I wasn't the only one.
No, it doesn't. It sounds like you definitely weren't. All right. Yeah, just so you guys know, definitely let me know when things like that are seem strange or don't seem right, because that means that there's more to it that I should have realized, right? That I didn't realize that there was some of these technical issues with that particular lab. So like I said, I'll look that over and take that into consideration when I'm entering your lab grades for that lab into Moodle.
Thank you.
Anything
Yeah,
else?
thank you.
You're welcome.
No, the only other thing is homework. For some reason, in my head, I had it mixed up that it was going to also be posted day later. I'm not sure where I got that from. I think I sent an email to the group email.
I can't. I can kind of hear you, but barely. So I'm not sure.
Is that better?
Yes.
Okay.
Thank you.
For me personally, the homework, I think for some reason I had it mistaken that you were pushing that up a day ahead as well.
Okay.
So of course, when I took it, it gave me 0 credit for it. So I'm
Right.
wondering how that translates on your end, because that
Oh,
was my mistake.
yeah, I can see the
Okay.
grade. I know it
Okay.
translates it as 0, but I can see what grade you got. So
Okay,
I'll
cool.
give you the grade for it, because
All
I
right.
know
Well,
there
that was
was
my mistake, but
confusion this week with the fact that I didn't finish the lymphatic system, and so I
right.
pushed
But the
homework
homework
back.
still, I should have still realized that it was, because it's always due the same day. So that was just a mix up on my part, but
Right.
I appreciate that. Thank you.
You're welcome. All right. So we were talking about lymphatic system. We were talking about adaptive immunity, right? Adaptive immunity is not what you're born with. That's innate. Adaptive immunity only happens when you see a certain pathogen and then your body creates immunity to it. And there are kind of these 4 properties of adaptive immunity, and that's where we left off. Adaptive immunity is always specific. It's specific, again, it's activated by a specific antigen, and your T cells and your B cells have receptors that only bind to that specific antigen. So whatever is causing that particular infection is triggered by a pathogen which has a marker on it called an antigen, and you are very specifically attacking that. So in other words, if you have chickenpox, your body is very specifically attacking varicella zoster because that's what's causing your chickenpox. It is versatile. There are millions of different lymphocyte populations produced, each with a different antigen receptor. So what your body does, which is really cool, your body makes up as many different T cells and B cells as it can, with each of them with different receptors. And the receptors are specific for antigens that you may encounter in your life, right? So you might be born with whatever, 1 million T cells. They all have different receptors based on the fact that you might encounter something that fits that receptor. only when you do actually encounter something with that receptor does your body clone those cells, those T cells and B cells, with that specific receptor. And then you end up with an army of T cells and B cells with that specific receptor. So, you know, there's the possibility of being, of having immunity or having adaptive immunity to really almost like millions of different pathogens and different antigens. And there's versatility in that you can actually trigger, the T cells to produce more of the T cells with that particular receptor through something like a vaccine, right? So a vaccine will actually cause your immune system to make up an army of those cells that only have that one particular receptor, that receptor that's specific for whatever the antigen is. Another really amazing thing about our adaptive immunity is that we have memory. The first exposure to anything, to any antigen, will clone those cells, clone those B cells and T cells. And part of that cloning is that they make memory cells. And memory cells last for years. Now, they make the second exposure to that same antigen a much faster, stronger response, right? So that's why you can't get chickenpox twice, because you already have an army of cells ready to go against that particular virus. And so you can't actually, you will get the virus again, but your body will fight it off so quickly and with so much strength that you won't get any symptoms of chickenpox. And then tolerance. Tolerance exists when immune system does not respond to normal antigens. So our immunity has this ability, our adaptive immunity has this ability to recognize our own cells from foreign cells. So there's an antigen on everything, right? There's an antigen on our own cells, there's one on foreign cells, and our immune system can tell the difference. So our immune system is supposed to leave our own cells alone and only attack cells that have antigens that are different than the ones on our own cells. And so here's the overview of adaptive immunity. So adaptive immunity is where antigen presentation triggers an immune response by specific defenses. All that means is that whatever is infecting our body has a little marker on the outside of it, and that little marker is called an antigen. So there are little red circles on this guy here. There are little blue circles over here, right? Those are the antigens, and those will bind to a specific receptor on a T cell or a B cell and trigger the immune system. Now, it can trigger T cells, which are up here, cell-mediated immunity. And when those are triggered, they activate the B cells. Or the antigen might trigger a B cell, might get recognized by a B cell, and the B cells can trigger T cells, right? That's why these two arrows are here. So your body's not just going to only have cell-mediated immunity or only antibody-mediated immunity. Your body wants the most immunity possible, right? So it's going to trigger both. And here it just tells you a little bit about what they do. The T cells, cell-mediated immunity is T cells, those can directly attack whatever the pathogen is, right? So they bind to that antigen and they can attack this bacteria cell or this virus, right? Or this virus. Whereas antibody-mediated immunity, which is B cells, the B cells actually circulate antibodies, and antibodies are these little Y-shaped proteins here. And what those proteins do is they bind to the antigen. So instead of the antigen being able to bind to another, like a host cell, to do whatever damage it needs to do, these antibodies bind up these antigens, so the antigens can't bind to anything. Okay. And then other white blood cells come along and they see these antibodies bound to these antigens, and they can chew up or engulf that complex. So antibodies, B cells, just kind of target these pathogens for destruction, whereas T cells can actually attack these cells. They can dock next to these cells and kind of push enzymes into these viruses and into these bacteria and kill them.
Can I ask something about this picture right here?
Sure.
The one that you just showed. Is this illustration is a first or second encounter? Is that
This happens on the first encounter. The biggest difference is that with the first encounter, from adaptive immunity to triggering these two processes takes like 10 days, right? So the
Okay.
first encounter, even the first encounter with the antigen, you're going to trigger the immunity, and it's going to take you about 10 days to your body, about 10 days to kind of clone everything, get an army ready to go and fight. Part of that process, if that happens with the first encounter, is that you make memory cells. And so if you get a second encounter, within hours, you have an army ready to go because you already have those cells cloned, right? And they're ready to go and they get mobilized very quickly. So this would happen even on the first encounter. It's what the difference would be is in this gray arrow here, it would be the time. If it was the first encounter, to get from the first box on the left to the second and third box would be about a week and a half. If it's the second encounter
Okay.
or past that, it's only going to be a few hours.
okay? So the second encounter is a lot faster,
Much faster,
right,
again,
because-
much stronger.
right? Because it's already built up. Okay, that's what I am trying to like separate what happening on the first encounter.
Right. And that's,
Okay.
we'll talk a little bit more about that in the next few slides.
All right, thank you.
You're welcome. So how are these T cells activated? Well, they're activated because they have these receptors on them, and the receptors are really specific to only one antigen, right? So you have a different T cell with a different receptor. It's slightly different for every single antigen that you might come in contact with. And so they have to recognize that antigen. One of the key things that T cells do is they can recognize antigens on your own cells versus antigens on foreign cells. The antigens on your own cells are called the MHC complex, the major histocompatibility complex. Okay? The MHC are the antigens that are sticking off of your own cells. And so T cells recognize those and they say, well, we'll leave that alone, right? I have type A blood. So I have the antigen for the A protein on my blood cells. My immune system stays away from that. It says, okay, those are normal. Those are supposed to be in your body. There are different classes of MHC proteins. We're going to get into.
Can I ask a
So-
quick question?
Yeah.
So for those who have organ transplants, is the MHC proteins involved in like anti-rejection drugs somehow? Like when it actually does work?
So two
Like,
things.
trick
So
your
if
body
you
into
get an
believing-
organ transplant, you have to, the person that donates the organ, right, they have to be a really close match. So
Mhm.
that's what they're looking at, is the MHC on the donor and the MHC on the recipient, right? Are they really, really close?
Yeah.
And then usually when somebody gets an organ transplant, they're also given drugs that suppress the immune system.
Oh, that's
So
what
it's
those drugs
it's
are.
not, yeah, it's
I see.
so again, it's great because it can prevent rejection, but it also leaves the person more susceptible to any
Yeah.
other infections, right? Because they're suppressing the immune system.
And that's so that the immune system isn't so ramped up
Right.
to,
That it
be
won't
able to
reject. Hopefully
recognize-
the organ will
yeah.
take, and then,
Yeah.
then everything will be okay.
Wow, that's interesting. I didn't realize that. Yeah, to be like more activity would maybe pick up on, you are foreign. Yeah.
Right. Exactly.
Wow,
Exactly.
I didn't realize that. Thank you.
You're welcome. So there are these class 1 and class 2 MHC proteins, right? MHC proteins are on every cell. That's class 1. They're just antigens. They're antigens on our own cells. They're called MHC. MHC2, class 2 MHC proteins are on antigen-presenting cells. Antigen-presenting cells are really cool. You have white blood cells, like macrophages, which are very nonspecific. They go around and they chew up things that don't belong. So they go through your body, they see something that looks like bacteria, they chew it up like Pac-Man. Well, that bacteria had an antigen on the outside of it, right? And so what a macrophage actually does is when it chews up a bacteria, it doesn't chew up the antigen. It saves the antigen and it puts the antigen on the outside of it, right? And it does that to show the T cells, hey, I just killed this thing. Look out for this thing, right? I just killed this thing that had these markers on the outside of it. So look out for them, right? So if we go back to this picture, let's say a macrophage came across this bacteria that's here. This pink oval is a bacteria. And it's got these red circles that are the antigen. The macrophage can chew up the bacteria, take those red circles and hold them up, put it on the outside of itself to show all the T cells that, hey, look out for anything that has this particular antigen on the outside of it because it's not supposed to be here. Okay, and that's how T cells are activated. T cells have to be exposed to these antigen-presenting cells, right? So APC stands for antigen-presenting cell. And all they are white blood cells that when they chew up, when they non-specifically chew something up that doesn't belong, they take the antigen from the outside of that thing they chewed up, and they put it on the outside of themselves, right? And the reason that they do that is just to kind of flag the T cells, to say, hey, T cells, look out for this. Anytime you see this red circle, that's something bad. That's something you need to go after.
In the crash course, it said something, he said something like, you know, it proudly wears its previously murdered parts and wears them proudly or something like that. It was
Exactly.
the way you described it.
Yeah,
Yep.
exactly. That's exactly what it's like. It's
Yep.
like taking almost like a trophy or something,
Yeah,
you know,
yep.
like warriors would do.
So will the body then attack that presenting cell ever because it thinks like, oh, that can it mistake that for the actual like thing that had the antigen?
Yeah. But that's not such a big deal, right? Because you have millions of, because those antigen presenting cells are non-specific white blood cells that are going through your body. And you have
Okay.
millions of them, and only a few of them are going to be holding up this antigen to the, you know, to the T cells.
Okay.
So yes, it can attack that particular macrophage, but it's not going to attack all macrophages. It's just going to attack the ones that have that flag on them. So that's the sacrifice, right? That's fine. Your immune system's okay with that.
Okay. Is that not part of like autoimmune disease that happening?
So no, well, autoimmune disease, what's happening is your immune system is not recognizing this MHC complex, right? So
Okay.
normally your immune system recognizes that the MHC is those are your own antigens,
Mhm.
and that's different than foreign. And an autoimmune disease, your immune system is seeing this MHC complex as a foreign antigen and attacking it.
Okay. So it's not about it presenting anything. It's just in general, it recognizes it wrong. Okay.
Yes. Yep.
All right. Thanks for clarifying that.
Yeah, you're welcome. So when the T cells see this antigen presenting cell, which is holding up an antigen saying, hey, I just killed this thing, they get activated, right? That activates the T cells and that initiates the immune response, which just means your body starts cloning more and more T cells that have the receptor for that particular antigen, right? Because now you need an army of T cells to fight off this antigen that's the red circle in this case. So T cells themselves, they have membrane proteins called CD, cluster of differentiation markers. And there are different markers on T cells, and that marker determines the response to the self-antigens on your own cells. So you've probably heard of CD8 and CD4 if you've been in the medical field. CD8 cells have a CD that's labeled as 8. Those are cytotoxic T cells. And so they respond to antigens on class 1 MHC proteins. And helper T cells, which are CD4, respond to antigens on class 2 MHC proteins. So this just deals with the fact that they are looking at these antigen presenting cells differently. Basically, CD8 cells, which are cytotoxic T cells, same thing, interchangeable, they are responding to antigens on class 1 MHC proteins, and CD4 cells, which are the same as helper T cells, can respond to antigens which are on class 2 MHC proteins. That's all. It's just a technical difference. And the T cell must bind to a second site for co-stimulation before being activated. So again, you need this antigen presenting cell. It's not like, oh, the bacteria is floating through your body and your T cell is automatically going to recognize it and then, trigger immunity. It needs to be presented with it. Something needs to kind of like tap it on the shoulder and show it this antigen and say, okay, you need to clone yourself and go after this. And then when the T cells are activated, right, whether it's CD4 or CD8, if it's CD4, it activates CD8, and if it's CD8, they're active. It makes an army, it clones an army of cells, and so you get CD8 cells, you get CD4 cells, you get memory cells, and you get suppressor T cells. You get this whole army ready to respond. Now, again, that takes over a week for that to happen. Right, it takes probably 7 to 10 days, they estimate. So what are these T cells? Cytotoxic T cells are sometimes called killer T cells. They are CD8 cells. When they become active, they divide into more cytotoxic cells and memory cells. They can destroy any bacteria, fungi, or even transplanted tissue, right? If they don't recognize the markers on that transplanted tissue. One of the things that they can do is they release this enzyme called perforin, and it actually perforates this target cell's membrane. It pokes holes in the target cell's membrane. And so water rushes into that target cell and it explodes, basically. They can also secrete these messages called cytokines. And cytokines initiate apoptosis. Apoptosis is programmed cell death. So they secrete these cytokines into the cell. When those cytokines get into this target cell, they cause that cell to die through a bunch of other mechanisms that we won't worry about. And they can secrete these lymphotoxins, which again disrupt the target cell metabolism. So if the target is something like a bacteria cell, it's going to mess with the cell's ability to make energy and that sort of thing, which is going to kill the cell as well. So the big thing is these cytotoxic T cells, which are CD8 cells, can go out and actually actively kill and destroy different cells, whatever these different pathogens are.
Are killer T cells and natural killer cells different?
Yes, they are different.
Okay.
Natural killer cells don't have to be presented with an antigen. Natural killer cells are not specific.
Okay. Well, natural. That makes sense. Okay.
Yeah. So here you can see activation of cytotoxic T cells, antigen recognition. So here is on the left on the right is a cytotoxic T cell, and it's got a receptor, and the receptor is going to bind specifically to that antigen. This cell on the left is a white blood cell like a macrophage, and what it's done is it's chewed up some sort of pathogen, and the only thing that it didn't chew up are these little red antigens. These were on the surface of whatever pathogen it chewed up. So this cell, this macrophage, goes ahead and it puts these little red markings these antigens on the outside of it, right? So it's flagging this T cell saying, hey, you know, I just killed something that had this on the outside of it. Come over here and check this out, and it activates that T cell. Now this T cell is active. Now its receptor specifically bound to an antigen. So now you're going to clone, your body's going to clone more of those T cells with that same specific receptor. Okay, and so these are the active T cells. At the same time, it's cloning memory cells that'll stay around for a long time. Now, you've got a bunch of these active T cells roaming around your body with the correct receptor. So anytime they see a cell with that particular antigen on the outside of it, they're going to connect to it, right? They dock to it, and they secrete these things into it, right? So here you can see in blue, this is the T cell with the receptor. This is the target cell in pink, and it has the little pink marker on the outside. The T cell is going to actually secrete things into the pink cell to make it die. Now, we also have helper T cells. Do helper T cells sound like they can kill anything?
No.
No, they can't. They're also called CD4 cells, right? Cytotoxic ones were CD8. These are activated by exposure to antigens bound to a class 2 MHC proteins. They divide into memory and active helper T cells. Helper T cells can activate cytotoxic T cells. They can't actually go out and do the killing themselves, but they can activate the ones that do. And they can secrete cytokines, so cytokines can go out there and do some damage. But their main role is they stimulate cell-mediated and antibody-mediated immunity. So what's going to happen with a helper T cell is the activation happens kind of the same way, right? This first step, instead of this cell on the right being an inactive cytotoxic T cell, it would be an inactive helper T cell. Then you would clone a bunch of helper T cells. Now what the helper T cells can do is they can go out there and activate cytotoxic T cells. So a helper T cell can act just like this cell. It presents the antigen to the cytotoxic cell, right? It says, look for this receptor, look for this particular antigen. It also will activate B cells, which we haven't talked about yet, but it's going to activate B cells as well, so that you're getting the best combination of both T cells and B cells fighting off whatever this infection is. Okay, so it stimulates both cell-mediated immunity, which is T cell, and antibody-mediated immunity, which is B cell. And then whether you activate cytotoxic T cells or helper T cells, you always create some memory cells when you have a cloning event. And those remain in reserve. And so what happens is if you are exposed to the same antigen again, they will rapidly differentiate. So you have an army ready to go. So the first time you're exposed to a particular antigen, it takes 7 to 10 days to get all this up and running in your body. The second time you're exposed, it takes less than a day. It takes several hours to get this up and running because you already have this army in reserve ready to go. Okay, and the fourth type of T cells that you make are regulatory. They are activated slowly by exposure to antigens bound to class 1. They suppress the response of other T cells and B cells. So they limit the degree of immune response to a single stimulus. The reason for that is that generally what makes us feel sick is actually our immune response, right? That makes us feel sick. It gives us the symptoms of being sick. So you need to have some regulatory T cells in there as well to limit that response to a single stimulus and to make sure that you're still responding to other stimuli as well that might be trying to infect your body. Now that's all T cell stuff, right? That's all cell-mediated immunity. Well, B cell activation happens kind of the same way. Okay, you have your body creates all these B cells that have different receptors on them. And when a B cell comes in contact with an antigen that's specifically binds to its receptor, you clone those B cells, right? And you have an army of those B cells that go out to do their job. So each B cell type has a specific antibody molecule in its plasma membrane. It has a specific receptor. When matching the antigen appears, so you've got a B cell, and on its surface sticking off is a receptor, and it's got a very specific shape. When that receptor binds to an antigen, then it goes ahead and it clones more and more B cells with that specific receptor. It also triggers helper T cells, which come back and clone more B cells, right? There's all this crosstalk between B cells and T cells. The activated B cells, right? They get activated, they clone themselves, they become plasma cells. So they become these cells that are in the blood, they're in the plasma, and they're secreting large amounts of antibodies. And then you also get memory B cells. Now, what you have here in step 1 is an inactive B cell. On the surface, you have receptors, right? Those receptors are the actual antibodies. So your body makes up millions of different B cells that have different receptors for any antigen it could come in contact with. If a B cell comes in contact with an antigen, right, such as this red circle antigen, and that antigen specifically binds to that receptor, which is the antibody, it sensitizes the B cell. That's just the terminology that's used. It activates the B cell. And what happens once they're activated, the B cells make an army. Now, the B cells do other things, right? The B cell can actually activate the helper T cell, which goes ahead and activates the T cell response, right? And that's what this is showing. So that if B cells are active, so are T cells, right? You're not activating just one or the other. You get both of them. And what's going to happen is you clone all these activated B cells. Now, some of them are in the plasma, right? They're in the blood, and some of them are memory cells, which are inactive, but they stick around. The ones that are in the plasma start producing more and more of the antibody. They're producing this receptor, right? And they're releasing that receptor into your bloodstream. Now, what this receptor does is it goes and it binds up that antigen, that red, in this case, that red circle antigen, so that whatever is carrying this red circle antigen can't do anything, can't do any damage, because it's all bound up by these antibodies. So the B cells don't actually kill any other cells like the T cells do, right? But they secrete these antibodies, which basically mess up the plan of the antigen, right? They get in the way of the antigen doing its job. And what's going to happen is you're going to get agglutination, which we saw with the blood testing, right? With the blood testing, when you took a drop of blood and you put it on that card had the antibody on it. So if you saw agglutination, if you saw clumping, what that meant was that there's a bunch of antibody binding to a bunch of your red blood cells, right? So in the case of mine, a bunch of antibodies that were anti-A antibodies bound to the red blood cells, which meant that the red blood cells had A's on them. So you get that clumping up, and now those red blood cells can't do anything because they're clumped up by this antibody. So again, these are cells that participate in defense. You've got cells that participate in tissue defenses, the cell and the function, neutrophils, eosinophils, mast cells, basophils. These are all non-specific. The antigen presenting cells can be macrophages, right? Again, they're non-specific, but they can chew up something and take the antigen and put it on the outside of themselves. And then you've got lymphocytes. Lymphocytes are T cells and B cells. So we've got, you know, the different types of T cells that we talked about, cytotoxic and helper and regulatory. We've got B cells. You've got memory cells, which can be any of these three types. All of this kind of gets activated at once, right? Your body wants to activate as much of this as possible. And so here is kind of a web, if you will, of what is going to happen with your body's immune system. So up at the top here, you have different pathogens. A pathogen is anything that causes an infection or a disease. So you have this pink one is bacteria. These other two look like viruses. They can trigger either innate, non-specific immunity or adaptive immunity. And if they trigger innate immunity, oftentimes there are things with immunity that go ahead and trigger active immunity. Right? Your body doesn't want to leave things up to chance. It wants to fight with as many different weapons as it can. Okay, and so within innate immunity, we talked about complement system and natural killer cells, macrophages, right? And they can, some of these can directly attack cells, destroy the antigens. Some of them can attack by circulating proteins and destroy the antigens. Some of these can actually go ahead and trigger the adaptive immunity, which leads to either cell-mediated or antibody-mediated immunity. Right? This is cell-mediated is T cells going out and having direct physical and chemical attack of the antigen. Antibody-mediated is T cells activating B cells, and the B cells secrete antibodies, and the antibodies lead to the destruction of the antigens. So you're getting all of this. If you look at the end, all of these arrows are coming into destroying the antigens. You're getting all of these things cross-talking at the same time, which is why it's confusing, right? But it's also why it works really well. Questions so far? I mean, that's kind of something you have to think about for a little while. Now this gets into a little bit more of what Juliet was asking, primary response and secondary response. The primary response happens the first time you see any antigen. It's your body's first exposure to any antigen. Secondary response happens after the second exposure or any subsequent exposure, third, fourth, fifth, whatever it is. So the primary response is the initial response to an antigen. The antigen has to activate B cells, which differentiate into plasma cells and memory cells. The antigen has to activate T cells. This antibody secretion, right? Those B cells, once they're active, they have to secrete the antibody, which kind of sequesters whatever's attacking. All of that takes 1 to 2 weeks to peak, right? So if we look at the time on the bottom of this chart, the primary response really peaks close to 2 weeks, something like 10 or 12 days. But the secondary response, you already have memory cells. You already have these memory cells cloned. And so all that happens is those memory cells, which are cloned, start putting out antibodies right away. And they can even clone themselves even more so that you get more memory cells. So with the second exposure, you can get a much higher response. First of all, look at how much higher this is than this, right? And it happens within a few days, sometimes even less than a day. So even within less than a day, you're going to get to whatever that level was of the primary response. Within a few days, you're going to get this incredibly strong response. And so the result of that is the secondary response would mean that you don't even feel the symptoms of it. You probably wouldn't even know you were sick because you wouldn't have any symptoms. Now, the theory behind vaccines and why they work is what the vaccine does is it triggers the primary response. Okay, so like COVID vaccines, a lot of us have gotten a COVID vaccine. It's triggering that primary response, which is why some of us feel sick after it. But what's going to happen now, hopefully, if all goes right, and there's other reasons to think that it might or might not happen, but the whole point of it is that the second exposure to that coronavirus will trigger this response. right? And so we won't actually get COVID. We won't actually get sick from it.
I have a question.
Sure.
So, you're talking about COVID and how the second response goes.
Mhm.
What is the third? I'm just trying to imagine what the third introduction of artificial immunity looks like after the second. Is it the same or?
Yes, in theory, it's the same. Now, what the wild card is that we don't know what the antigens on the outside will look like, right? So okay, all this is based on these antigens that are on the outside of the virus, right? These little blue things that are on the outside. And so the vaccine that's made is made in response to these antigens, right? But they haven't
Right.
sequenced the entire antigen and part of the antigen. So it triggers the immune response. But if the part that they've sequenced ends up being a little bit different the next time that virus comes around, then it may or may not trigger as strong of an immune response. It's the same problem that we have with the influenza virus, right? Those proteins that are on the surface, those antigens actually change and they look a little bit different the next time around. And so you have immunity to this very specific looking one. And if it looks different the next time around, just a little bit different, are you going to have that same immunity to it, right? And that's kind of the unknown right now.
Okay, so the variation.
Right. So, and that's just random, as time goes on, things mutate, things change, so that the variation is that's one reason why people are skeptical of it.
Right. I was just wondering, what it looks like because of they're talking about boosting the vaccine. It's like, okay, so what it looks like, is it the same as the second?
Well, in memory cells too, some memory cells stay around for years and others don't, like tetanus, right? You have to get tetanus boosters every 10 years or so
Right.
to make sure that you still have this high secondary response. And that's just because the memory cells are not necessarily going to remember that particular antigen. There are other cases where, it can stick around for a long time. I don't know how many of you guys remember like bird flu that came around, 10 years ago maybe? They expected the elderly population to be significantly affected by that flu virus because their immune systems are weakened, and they found that they weren't. That younger people were actually getting sick with it more often than elderly people. And it turned out that there was that exact variant came through like 70 years before that. And so people that were
Wow.
elderly that were alive 70 years before that still had memory cells against it. And so that was a case where the memory cells really did last a long time. They really did last 70 years, So I don't know. I mean, different antigens can trigger a different response. So there might not be memory cells left in some cases.
It's just interesting. Thank you.
You're welcome. Okay, so then we get to situations where the immune system doesn't do what it's supposed to do, right? A normal immune system has immunological competence, right? If somebody's competent, they're good at what they do. So this is the ability to produce a normal immune response after exposure to an antigen. So you see an antigen, it causes it can cause a disease. You have a normal immune response. But there are people who have there are plenty of people that have disorders, such as autoimmune disorders or immunodeficiency diseases or allergy diseases, where things don't go the way that they're planned. With autoimmune disorders, what's happening is that the immune system mistakenly targets normal body cells and tissues. So normally, an immunocompetent system distinguishes between those antigens that are self, right, those MHC antigens, versus foreign. But if you have an autoimmune disorder, your immune system doesn't, right? So it thinks that something is foreign when it's not. RA, rheumatoid arthritis, is a very common autoimmune disease. The antibodies attack connective tissue around their joints, right? And people get arthritis. Type 1 diabetes mellitus, which is very common. Some people are born with it. The antibodies are destroying particular cells in the pancreas that make insulin, called islet cells. And so the immune system is somehow recognizing those as something foreign, even though they're your own cells, and it's attacking them. And so you can't make insulin, and therefore you have diabetes. So it just appears to be a case of mistaken identity. There's a lot of really cool research going on with this. Because so many people have rheumatoid arthritis, a lot of drug companies really want to get things on the market for that. And so there's a lot of really interesting research that's maybe not all of your cells have exactly the same DNA, right? Which we always thought for a long time they did, but maybe they don't all. And so there could be a reason why your immune system is recognizing those cells as foreign if they're slightly different than the other cells in your body. Immunodeficiency diseases is usually where the immune system fails to develop normally, or it's somehow blocked. So people that are born without an immune system, or who don't develop an immune system, the term for that is SCID, which is severe combined immunodeficiency disease. And those of you that have any experience with research in mice, a lot of times there's a lot of mice that are produced as SCID mice so that you can do different research and they don't produce an immune response, so you can see what the progression of the disease is. Obviously AIDS, which is something I think everybody has heard of. AIDS is acquired immunodeficiency syndrome, and it's a result of the viral destruction of helper T cells. So AIDS is caused by a virus called HIV. HIV targets those T cells. It targets helper T cells. And if you remember, helper T cells activate cytotoxic T cells and B cells. And so all of a sudden, if you don't have helper T cells, if they're getting killed, then you don't have an immune response. And people with AIDS usually die of a secondary infection. They usually die, from some other infection that most of us can fight off with no problem, that their body can't handle because they don't have the T cells to do it. And then allergies. Most of us have experienced allergies. Allergies are inappropriate responses to antigens. Hypersensitivity. Basically, your body thinks something is dangerous when it's not, and it overreacts, right? And that makes you feel sick. So I'm not going to get into all these different, type 1, type 2, type 3, type 4 things. Type 1 is the most common. It's what we've all experienced, right? It's springtime now, things are blooming. We all have this experience of having the pollen in the air, and it makes us feel crummy. Basically, your immune system is overreacting to the pollen, to the antigens on the pollen themselves, and producing more histamine, producing more mast cells, producing more white blood cells to try to combat that when it's really not anything that's dangerous. This is, same thing with, some people are allergic to peanuts. That's a common one. Bee stings, right? Those things are pretty common. That's just an oversensitivity to things that you don't need to be that sensitive to. And more severe types of allergies obviously are, things like kidney diseases, right? Immune complex disorders where there can be certain materials going through your kidney that are getting filtered out of your kidney that your body somehow recognizes as foreign, and then inadvertently attacks the kidneys because of it. But those are much more severe, and those are usually more rare.
So anaphylactic shock would just be like an accelerated amount of what you were just talking about with an allergy? Or is
Yep.
that
So
okay?
yeah, that's an allergy. That's like a type 1. It's an immediate hypersensitivity. So what your body does is it overreacts, and it produces a bunch of inflammation really quick.
Yeah.
And what happens is the inflammation inflames your throat, your trachea, and all of a sudden you can't breathe.
But that's from the histamine.
Yeah.
Okay. as
That's
well.
from your
correct?
immune system.
I didn't
Yeah.
know if anything in addition to that or something happened because of the severity of it, but it's just that.
Nope. Yep. It's just your
Wow.
body's response.
Wow. Okay. Thank you.
Welcome. So what kind of questions do you guys have after that? And I think you can see now why I didn't want to rush the rest of that, because it's lengthy. There's a lot of different things in there.
I can see some parallel or like connection between this allergy pathway and like autoimmune pathway. Are they directly related? Like, can allergy lead to the other because of the like hypersensitivity?
Not that anybody has found. It's usually that the autoimmune diseases, it's that cells from one particular part of the body have a different antigen on them,
Mhm.
or a slightly different antigen, and so that's what your body is reacting to. Like for instance, Crohn's disease attacks the digestive system. Um, there's evidence, like I said, more evidence now that not all of our cells will have exactly the same DNA.
Hmm.
And so, what does that mean? Does that mean that, that if you have slightly different DNA in a certain region of your body, that you're going to have a different antigen on those cells, and your immune system is going to recognize that differently? And that may be the case. That may be what's going on.
Is that also why somebody could develop, say, like a peanut allergy in their adult life when they never had a problem when they were a child?
No, that's usually, so an allergy development is usually just exposure to it after a number of times. Sometimes that triggers the immune system to have a stronger response to it.
Yeah, I've
Um,
always been confused by that. That's the same with like lactose, right?
Well, no, lactose is actually being lactose intolerant is completely normal
Yeah, we talked
as
about
an
that
adult.
before.
Yeah. So, and that's different. That's not an allergy. That's just that your body doesn't make the enzyme anymore to break down the sugar in milk.
Oh, okay. I thought it
So
was
most people as adults don't make that enzyme. That's
Oh, okay.
the normal thing, is that the wild type, I guess is what it's called, is
Yeah.
that you wouldn't have any reason to break down milk sugar, because you wouldn't be drinking milk unless you were an infant.
Right.
So that's where lactose intolerance comes.
It's just so strange because it's like, why would you develop an allergy after being exposed to something so much, like and not have
Right.
a problem. It's really confusing, but
Well,
yeah.
I mean, similar thing with some people develop diabetes, type 2 diabetes, right? So they
Oh, that makes
overexpose their
sense.
body to sugar,
Yep.
and eventually the body reacts to it differently, because you've just exposed yourself to it so many times. The body starts saying, okay, I'm going to react differently to it.
Well, putting it in that perspective, actually, that makes sense. Yep.
Yeah.
And I guess is that the theory of allergy shots, like expose the body so much or the
Yeah.
immune system so much that then it's a normal thing?
Yep.
Okay.
Yeah, I went through allergy shots for about 6 years when I was a kid.
I went through them too for a few years, but didn't seem to help that much. But they can really be beneficial, so I'm not saying for
Right.
anyone that they shouldn't do them, but yeah,
Did you go through it as an adult or as a child?
as an adult, actually.
Yeah. See, I think they're more beneficial as a child, because like all this stuff that we're talking about, your immune system is still developing, right? I went through allergy shots from the time I was like 9 to 15. And so, it really helped me. I really don't get much seasonal allergies now compared to a lot of people around me. And I think that that's a big part of it. I think, your immune system is shaped in that time frame. But
Yeah,
getting those tests is no fun. The shots all over the back to see which ones will react.
yeah, that is not fun. Lots of itchy, scratchy stuff, but
Right.
What you're saying about like how they're recognizing that some human cells are a little bit different and maybe not totally identical. It's really fascinating. Like maybe, I guess it's that bigger question too of like, can the immune system adapt, like coincide and adapt to those like minor changes, or how much is like too much of a change? I'm very curious about that.
Yeah, there's so much. There's all kinds of new studies too on the bacteria flora that live in your gut. right? We all have bacteria that live in our digestive system that help us digest foods and everything, and help us stay healthy. And there's a lot of evidence that some people who have digestive problems don't have the same bacteria in their gut. And so what they're doing is they're actually taking bacteria from healthy people's guts and putting them into, you know, putting that into somebody who's sick all the time, and seeing really, really good results with that. Like, ridiculously high numbers of success with that.
Wow.
Yeah.
Yeah, it seems like so many things, disease and otherwise, come back to the gut. Like at some point, they just trace back to gut health.
Absolutely.
Can I ask something?
Sure.
What is idiopathic allergy? My son was diagnosed with it, and I don't understand what it means.
So idiopathic, that's just a big word for them saying they don't know why.
Oh,
It's a horrible thing. Idiopathic, whenever doctors use idiopathic, whenever you see it in the biological sense, it literally means that the cause is unknown. So what happened is they probably did a bunch of tests and couldn't figure out, narrow down, oh, it's not this, it's not, it's not A, it's not B, it's not C, but we don't know exactly what it is. Like, they didn't get a positive result saying he's allergic to this, right? So was it food, or was it like a skin allergy, or like sometimes idiopathic a lot of times can be, we don't know if it's your detergent that you're using, if it's your soap that you're using, it's could be anything causing a rash on you, that kind of thing. Or sometimes with food, it's really hard to eliminate certain foods in order to see if that's the cause.
all right,
But yeah,
so they have no
idiopathic,
name.
I hate that term, but that term literally means they don't know what's causing it,
The
Yeah, I
root
did
word
a
is kind of funny
research.
too.
right?
That I think it's just fitting idiopathic.
Right, exactly.
Yeah, that's exactly what I was saying. Yeah.
Exactly.
All right, thank you.
All right, so the exam goes through the cardiovascular system and the lymphatic system, since they're closely related, since it's, we're talking about blood cells and T cells and B cells and all that. That's all on one exam. Now, that exam is due Monday. And what I want to do next is start on the respiratory system, which I feel is a lighter chapter. The respiratory and digestive systems, I actually kind of enjoy these two chapters a little bit more. So I want to start in the respiratory system, and that way we can discuss it a little bit more tomorrow, and we can discuss the respiratory physiology, the respiratory physiology lab for this week, which is cool. It's something that doctors often will do. If you're seeing a doctor, they'll try to figure out how much air you're getting into your lungs, right? And they use a machine called the spirometer. And it's something that I've done in labs a lot of times with students, but we can't do it right now because of COVID, because you have to blow into things, and that's not recommended right now. So I'm anxious to do it. And, I'm interested in having you guys do this and seeing what your numbers come out like. It's always, I don't know, I think it's always kind of an interesting thing to do. So let's switch gears to the respiratory system. All right. So the respiratory system. Why is it important? Let's go back to that. What's the respiratory system do for us?
Oxygen
Blood.
and dioxide.
Exchange of gas, oxygen.
Okay, good. It allows us to take in oxygen. Why is oxygen important?
Blood cells.
So, yeah.
ATP.
Mhm.
ATP, good.
Always goes
Oxygen
back to
is,
ATP. Yep.
yep, it all comes back to energy, right? All of our cells need oxygen and glucose to make energy. And so the way that we get oxygen is through our respiratory system. And we'll see that this is closely linked to the cardiovascular system. We have to get the oxygen into our lungs, and then from our lungs, get it into our blood, so our blood can take it all over our body. So that's the most important, I guess, role of the respiratory system, is getting oxygen into our body. Now, somebody else said CO2. That's absolutely right. Part of the way that we get rid of carbon dioxide is by exhaling it. And carbon dioxide is a waste gas. It's a gas that builds up inside our body that we need to get rid of. There are a few other parts of the respiratory system that are beneficial to us. For example, our sense of smell is linked to our respiratory system. That's in our nose. So that allows us to smell all these different odors, all these different chemicals in the air. Also, the respiratory system allows us to produce sound. We have our vocal cords. We produce sound by passing air across them. And so that's, a really important side effect for our lives, obviously, that we can produce this sound through our respiratory system. So the respiratory system consists of structures that are two things, kind of broken down into two types. There are some structures that are there to physically move air into and out of our lungs, right? Like our nose, like our mouth, and even the muscles that are surrounding our chest. And then there are structures that are there to allow for gas exchange. Then gas exchange is a term that means getting oxygen into our blood, right? So that our blood can carry it around our body, and getting carbon dioxide from our blood. Our blood actually carries the carbon dioxide around, brings that to our lungs so that we can exhale it. And so those two things are kind of the biggest parts of the respiratory system. So it provides a large area for gas exchange between the air and the circulating blood. This allows us to get oxygen into our blood. It moves air along the passageways to and from gas exchange surfaces of the lungs, allows us to get air into our body. It protects respiratory surfaces from dehydration, temperature changes, and pathogens. One of the really cool adaptations that we have is that we can live on dry land. And that's because our lungs are on the inside, and we can keep them from drying out, right? We can keep the moisture there. We can keep the temperature pretty constant. We can keep it warm. And we have white blood cells that we just discussed that can travel around in our lungs, like macrophages that can chew up things that don't belong. Now, that's an evolution thing, right? How do fish, what do fish breathe through? Do fish have lungs?
Gills.
Gills.
Fish have gills, right? And if you go out fishing and you catch a fish and you put it down in the boat, can it still breathe? No. Well, why not? The gills are right there on the side of its head, and there's all kinds of oxygen around, right? The reason it can't breathe is because the actual site of gas exchange, which are the gills in the case of a fish, have to stay wet. Okay, so our lungs have to stay wet. There has to be moisture there in order for gas exchange to happen, in order for oxygen to get into our blood. And the reason that fish can't breathe out of water is because their gills dry up, and then they can no longer get oxygen across their gills to get into their blood.
Because they're external, huh? I never thought of it that way. Wow.
Yep, exactly.
Yep.
And so that's, that was a big evolutionary thing that allowed for animals to proliferate in a dry environment. Number 4, produces sounds, right? We can produce sounds to speak and sing and communicate with each other. And that's because of passing air across our vocal cords. And also we have the sense of smell. The receptors for smell happen to be in our nasal cavity. And so that is a function of the respiratory system as well. Okay, as with most things in the body, we make up divisions in order to study it a little bit better, right? We compartmentalize everything. And so we divide our respiratory system into an upper and a lower, just to make it a little bit easier to study. The upper respiratory system is everything above your voice box, everything above your larynx. So your nose, your nasal cavity, your sinuses, your pharynx, which is the chamber behind your nasal cavity. And the whole purpose of the upper respiratory system is to not only bring the air in, but to filter that air as it comes in. And so we've got all kinds of structures in there that are going to filter the air. You also want to warm and humidify the incoming air. You want the air to be warm, and you want there to be some moisture associated with it. Again, if there's no moisture associated with it, and if it's not at a certain temperature, gas exchange is not going to happen at the rate that it should. So you'll see that we have structures in our nose, for instance, that look like swirls. And so when the air comes in, it swirls, and by moving the particles in that way, it warms them up. Okay, and so we're doing that to try to make gas exchange more efficient. The lower respiratory system includes the larynx, the trachea, the bronchi, and the lungs. Your larynx is your voice box. That's where your voice box is. So everything from the larynx down, right? The trachea is your windpipe, and then it branches into 2 branches that go into each lung, one into each lung, and then your lungs. And we'll see that the lungs contain smaller branches and eventually contain these little sacs called alveoli. So here are structures of the respiratory system. You can see that they've kind of divided these into upper and lower, right? So the upper includes the nose. There we go. The nose, the nasal cavity, the sinuses, which are behind the nasal cavity, and the pharynx. The pharynx is this open cavity behind your nose and your mouth, right? That's all upper. Lower starts at the trachea. Starts at the larynx. So above the trachea is your larynx. This is your voice box. This is your Adam's apple. Okay, trachea, which divides to go into each lung. Those first divisions are called bronchi. And then within the lungs, it branches into smaller branches, which we'll get into and we'll talk about. And eventually at the end of each of those branches are these little air-filled sacs called alveoli. Okay, so
Can I add something?
Yeah, go ahead.
Sorry.
No, it's fine.
Just the upper and lower respiratory. So when you're diagnosed with upper respiratory, it usually from, like on your chest or on the neck or something. So why is it called upper when it's technically anatomically lower?
So I think usually upper respiratory, like an upper respiratory tract infection or something, is going to be, an accumulation
Right
of
there.
mucus up here, there's an infection up here, that kind of thing. If it's laryngitis or if it's something in your lungs like COPD or asthma or anything like that, it's not considered upper respiratory.
It'd probably be
that's-
bronchitis or pneumonia if it was lower, right?
Right.
Yeah.
Are those, I don't know if doctors,
That's interesting,
doctors
actually,
call
because
those
they
upper
do say upper
respiratory.
respiratory. Yeah.
Because I have chronic. It's like if I've been exposed to cigarette, for instance, I will develop. It's like my throat will start producing a lot of mucus. Then I will have those laryngitis. It's all blisters, and then I will have fever, and it's like every month. Every time I expose to something, it's a chronic thing. And they said, oh, you have upper respiratory, but they're listening to my chest and I can feel it right on my throat, actually. It's clogged in there.
Right.
It's like, so why it's upper respiratory? I
So
don't
why
know.
it's not-
That's
yeah.
what I'm asking.
Yeah, I don't know. I don't know if medical doctors only consider it, like if it's in your lungs, that it's lower respiratory condition, like COPD or emphysema or something. I don't know. That's a good question. I don't know why it wouldn't go with these same divisions.
I wonder
Because,
if upper is easier to clear or something if it's in your lungs. I don't know.
Probably, yeah. As long as they said, as long as your lungs is clear,
Yeah.
they don't consider it lower,
So maybe they only consider it lower, like you said, like if you have pneumonia, if you have something that's, blocking something in the lung,
Right.
maybe that's when a medical doctor would call it lower. Maybe they would call anything in this part upper.
Trachea and larynx upper, yeah. That's why it's like, maybe that's how it goes. But now when I see anatomically, it's like, oh, I'm just triggered my curiosity.
Right. Yeah, I don't know why they don't go that way, or why.
Thank you.
I don't know who came up with that division first, but I don't know.
Medical doctor vocabulary, who knows, you know? Like,
Mm.
True.
has their own meetings, yeah.
Okay, so the respiratory tract is just the passageway that carries air into and out of the lungs. And there are two portions of it. There's a conducting portion and a respiratory portion. The conducting portion is just conducting air, right? It's getting air into your body and out of your body. That's it, right? So from the nasal cavity all the way down to the bronchioles, which are in the lungs, it's conducting air. You're trying to get oxygen in and get carbon dioxide out. And that's where, along the way, you're filtering and warming and humidifying the air. Once you get down into your lungs, you get to these smallest and thinnest little tubes called bronchioles, and you get to these little sacs called alveoli. And that's the respiratory portion. That's where you're actually getting oxygen into your blood and getting CO2 out of your blood, right? That's where the gas exchange is happening, so all the way down in your lungs. And so maybe this is more of the division that the doctor, medical doctors use when they say upper. Maybe they mean any of the conducting portion of your respiratory tract. That would be my guess. Okay, so the nose, right? We'll start at the nose. Air enters through the nostrils, which are called the nares. The space that's just inside your nose is called the vestibule. And then there's a nasal septum, which divides the left and the right nostrils. The floor of your nasal cavity, the bottom of it, is your hard palate, right? It's the same thing that forms the roof of your mouth. That's what divides your nasal cavity from your oral cavity. The soft palate extends behind the hard palate and lies under the nasopharynx. We'll get to that in a minute. The nasal cavity opens into the nasopharynx, and then you have nasal conchae. Nasal conchae are these swirl-like structures, so that when you inhale air, it's moving, and the reason it does that is to try to warm up the air. The nasal cavity is continually cleared and flushed. You have all these cells that produce mucus. So you have respiratory mucosa that produces mucus. The paranasal sinuses produce mucus. And you have tears flowing through your nasal lacrimal duct. The tears that go across your eyes, they empty into your nasal cavity. And so your nasal cavity is exposed to anything that you breathe, right? So dust, debris, allergens, pollen this time of year, right? Certain vapors, certainly if you decide to paint a room in your house and you don't have good ventilation, or even you clean the bathroom with a really strong cleanser and you don't have good ventilation, that can increase mucus production, right? That can make your eyes water, produces more tears, produces more mucus production too. The pharynx is also called the throat. And we're going to get to a picture of this in a second. It is a chamber that's shared by the respiratory and the digestive systems. It extends between posterior internal apertures and entrances to the larynx and the esophagus, blah, 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 big words. So the pharynx is subdivided into three regions: the nasopharynx, the oropharynx, the laryngopharynx. And I'm going to go to this diagram because it makes a little more sense. So there's a lot of things labeled on this diagram, but I want you to pay attention to just a few. Right, here's your nasal cavity, right? The nostrils, the air is coming in here. Those are those conchae, those are those swirl-like structures that allow the air to move as it comes in to warm it up. Behind your nasal cavity and behind your oral cavity, this is your mouth and your tongue, is one chamber. And this chamber is called the pharynx. So it's open to both your nose and your mouth. Now it is divided into three regions. Those three regions are the nasopharynx, the oropharynx, and the laryngopharynx. The nasopharynx is behind your nasal cavity. That makes sense. The oropharynx is behind your oral cavity. That makes sense too. Below both of those is the laryngopharynx. Okay, and that's open to your larynx. This is your larynx down here. There's nothing dividing this cavity, right? Those are just three regions of the same cavity. There's not actually anything that divides them. Now, here's the problem, right? Here's the problem that we have. At the bottom of the pharynx, the bottom of the laryngopharynx, it's open to two different tubes, right? It's open to the esophagus here, and it's open to the trachea, right? This is the
This is
larynx
where
and
the
the trachea.
whole wrong pipe thing comes in, huh?
Exactly.
Aha, okay.
So your esophagus goes where? Where would your esophagus lead to?
Your lungs.
Stomach.
Stomach.
Oh,
Where
yeah.
does your trachea lead to?
There we go.
Your lungs, right?
Yep.
So now the problem becomes, well, this chamber is open to my mouth and my nose. So I want air to come into this chamber and to go down the trachea to go to my lung. But I want food and drink to go to this chamber and to go down the esophagus to go to my stomach. So what your body does, which is pretty amazing, is your body, when you are getting ready to swallow any food or drink, this structure, which is light blue here, and it's called the epiglottis, covers up your trachea. And the reason it does that is so that food and drink only has an opportunity to go down your esophagus. It does not have an opportunity to go down your trachea, to go to your lungs.
Can I ask a quick question?
Yeah.
So I have a Huntington's disease resident who often aspirates food.
Hmm.
And I'm wondering if there's a problem with that, what you just mentioned happening because of his disease or something.
Mm-hmm. Yep. So,
Must be.
does the resident have like? So I'm not familiar too much with Huntington's. Is there a lot of muscle
Yeah.
loss
Yes.
associated with that?
Yes.
So, again, he or she, because of the muscle loss, the epiglottis probably, because there's muscles that control the movement of this.
Yeah. Wow.
Right. So if that epiglottis isn't closed, some of that food or drink could go down that patient's trachea to go to their lungs.
He goes and does
So-
barium swallows often to see the progression of his illness.
Gotcha.
I wonder if this is what they're looking for.
Yes. So
Interesting.
basically the muscles, like the muscles associated with the mouth, are the same muscles that cause this to close. Because what your
Right.
body does is your body knows when you're getting ready to swallow something, when you're chewing
Yep.
something in your mouth, and that's when this epiglottis is forced closed to cover up the trachea. So
Wow.
as this disease is progressing, that patient is losing more and more of this muscle, the ability to contract this muscle. And so some of that, whatever that person is eating or drinking, can go down their trachea as well.
Yeah. Wow.
Yeah.
I understand it a lot better now. Thanks.
You're welcome. Now the other amazing, so your body does that, which is pretty cool. This right here, this division between the oral cavity and the nasal cavity, that's called your hard palate, right? There's a bone there. If you put your tongue on the roof of your mouth, you can feel the bone. As you go towards the back of the roof of your mouth, it's called the soft palate, right? It's just tissue. It's not, there's no bone in there. When you are getting ready to swallow food, at the back of this soft palate, you have this little thing hanging down that looks like, it's not drawn here, but it looks like a punching bag, right? It's called your uvula. When you are getting ready to swallow food or drink, your soft palate and your uvula move back and up and completely block off your nasopharynx. So not only does your body cover up your trachea, but it blocks your nasopharynx. If you were to drink a big glass of water all down at once, you are holding your breath while you're doing it, whether you realize it or not. And the reason for that is because your brain doesn't want to have signals crossed, right? You don't want to be taking in air, which you want to go this way, and also drinking liquid, which you want to go the other way. So what your body does is it completely cuts off the flow of air, and it covers up the trachea so that food and drink only has one place to go. Now, oftentimes, we've all probably reached for a glass of water and you kind of hiccup or you start to sneeze as you're going to do it, and you suck in air. And so what happens then when you go to drink that water, some of it goes down the wrong pipe, right? And that's what we say, it goes down the wrong pipe. Well, what happens is if your body does inhale a little bit of air as you're trying to swallow, your body doesn't know how to handle that. Brain doesn't know how to handle that. And so some of what you're drinking or eating can start to go down your trachea. Now it's very, very, very tiny amounts because you're coughing it out. But that's when something goes down the wrong pipe. That's what's happening. And so in this way with the pharynx and the 3 different regions of the pharynx is related to the digestive system as well. So we talk about it, the respiratory system. You'll see it again with the digestive system because it has to do with how we get food and drink through our oral cavity down our esophagus.
I'm sorry, did you say the glottis or the epiglottis is the thing that stops the food from going towards?
The glottis. So I'm sorry,
glottis.
the epiglottis. The epiglottis is this this light blue flap here, which
Yeah.
comes down and covers up the glottis. The glottis is the
Okay.
opening, the actual opening of the hole. So the
Okay.
epiglottis covers it up.
Okay, thank you.
You're welcome. All right. So that's all upper respiratory system, right? You can see everything in here, the nasal vestibule, the nasal conchae. You can see where the sinuses are. You can see the nasopharynx, the oropharynx, the laryngopharynx. Everything from here down is considered lower respiratory. This wider area, kind of at the top of your trachea, is called the larynx. That's your voice box. That's where your vocal cords are, right? And that's what your voice box is. So the tube that surrounds and protects the glottis, right, which is your voice box, that's where the opening is called the larynx. Air leaves the pharynx and enters the larynx through a narrow opening of the glottis. Basically, I'll show you a picture of it, but that's where your vocal cords are, and as you pass air across them, they vibrate just like a guitar string vibrates, and it makes a sound, right? And then we shape that sound with the muscles of our mouth and our tongue and our lips and all that stuff. The larynx is made of 9 cartilages, which are stabilized by ligaments and skeletal muscles. So the cartilages keep the shape, right? They keep it open. They keep that airway open for us. The 3 largest cartilages are called the epiglottis, which is that flap that covers up your glottis, your thyroid cartilage, and your cricoid cartilage. We'll see these in a picture in a minute. And then there are 3 smaller cartilages, the arytenoid, the corniculate, and the cuneiform, which you don't have to worry about those. So the large cartilages, the epiglottis, is the one that comes down and it covers up the glottis when you swallow so that food and drink don't go down your respiratory tract. Your thyroid cartilage is the one that forms the anterior and lateral surfaces of the larynx. The ridge is what your Adam's apple is. So your Adam's apple is actually part of this thyroid cartilage. Everybody has an Adam's apple, men and women. The growth of that thyroid cartilage and the growth of your vocal cords is dependent on testosterone. And so when a boy hits puberty and he gets an influx of testosterone, their Adam's apple grows and their vocal cords grow. And that's why their voice deepens, is because their vocal cords are bigger, right? So like if you think of guitar strings, if you think of a bass guitar, the strings are thicker, they make a deeper sound when they vibrate. And that's why an Adam's apple is usually more pronounced in a man than it is in a woman, because a man naturally will have more testosterone in their body than a female. Just below the thyroid cartilage is the cricoid cartilage. It's a ring of cartilage just below kind of your Adam's apple, and that's just above where the trachea starts. So here's a picture of this. So from the front, here's your thyroid cartilage. That's your Adam's apple. That's the part that kind of protrudes from the neck. And then below that is a big piece of cartilage called the cricoid cartilage. And then there's your trachea with the cartilage rings. So in this drawing, everything that's light blue is cartilage. You can see the epiglottis at the top. The epiglottis kind of flops down and covers up the vocal cords. If we were to look at this from behind, right? And that's what this is, a posterior view looking at it from behind. They've opened it up, right, so that you can see into this larynx. And here you can see the epiglottis. The epiglottis the bottom of it would stay fixed where it is, and the top of it would flop down towards you, out of the screen towards you, and cover up this, this opening right here. So I lost my cursor. Here we go. This opening right here would get covered up by the epiglottis. It would flop down out of the screen towards you. And so you can see the back of the thyroid cartilage, the back of the cricoid cartilage, right? I'm not going to hold you guys to the other cartilages, the arytenoid, or the other two that they mentioned, the cuneiform and the corniculate. And so that opening that we pass air across, that's where our vocal cords are, right? We have what are called false vocal cords, which are up here. They don't move when air goes across them, they don't vibrate. But the true vocal cords, which are down below it, are right here. And what you do is when you pass air across like coming out of your body through your trachea, it causes these vocal cords to vibrate, and that's what produces a sound, right? And so here are the actual vocal cords. So when you're looking down somebody's throat, when they put a scope down your throat and they look at it, these are the vocal cords right here, okay? This one here and this one here. And right now they're separated, and that's that space between them, that triangular dark space between them would allow for air to pass through. Now, as you're making noise, as you're making sounds, these vocal cords are smashing together, okay? And the closer they are together, the higher pitch they'll vibrate, right? And the further they are apart, the lower pitch they'll vibrate. So here you can see up above, here's a drawing of it. Here's your true vocal cords, right? Here they are separated, here they are closed, right? So the glottis in the closed position like this, and the glottis in the open position. One of the things I should post on your Moodle shell, one of the videos that I used to show students in the classroom goes through a lot of these different things. And one of the things they do with this particular part is they put a scope down Steven Tyler's throat, and they have him sing while the scope is in his throat. And so you can actually see in real time his vocal cords smashing together as he's singing Dream On. You know, as he's hitting those high notes, you can see how fast they're smashing together. And it's pretty cool the way it illustrates, you know, the way it illustrates how that sound production works. And there's a lot of people, singers, you hear about it a lot, that damage their vocal cords. They have to have surgery done on their vocal cords, and they have to have, it's because the vocal cords smash together so many times with such force that people who speak for a living, people who use their voice a lot, often damage their vocal cords and have to have surgery to repair them because of the forces of them smashing together so often.
Is that what would cause losing your voice?
Laryngitis?
Well, no, I mean, like, I'm a football mom, so I scream
Okay.
heavily at my son's football games, and then I
Yep.
can't talk the next day.
Yep.
Is that why?
So what happens is, you know, all of us, like if we go to a concert or something like that, or a sporting event and you're yelling, you'll lose your voice the next day. What happens is these vocal cords, you overuse them and they become inflamed, right? And so
Okay.
when they become inflamed, they don't vibrate as much. And if they're not vibrating as much, you can't produce as much of a loud sound. So
Okay.
like laryngitis, it could happen with an infection that could cause them to get inflamed, right? And that would be something like laryngitis, where you can only get that whisper out. But the same thing, if you overuse them, then yes, it's because they're inflamed.
Okay, that makes sense.
Yeah.
Thank you.
You're welcome. And here you can see the epiglottis. This is in the up position, right? So this would fold down over the vocal cords when you're swallowing food or drink. So vocal cords produce sound production, it's vibration of air over the vocal cords. The pitch is determined by the diameter, length, and tension of the vocal cords, right? So If you have short, thin chords, it's going to produce a higher pitch. Long, thick chords produce a lower pitch. Again, just think of it like guitar strings, right? The thicker the guitar string is, the lower the pitch. The pitch is altered when the position of the cartilages is changed. So the arytenoid cartilage, which I'm not going to hold you to, but that's how we actually move our vocal cords. And so you can kind of stretch them to be longer and thinner, or allow them to be, you know, looser, and so they'll be thicker, and you'll change the way that they resonate. But all that does is produce the sound. It's what you do with the sound once it gets up to your oral cavity and your nasal cavity that allows us to speak or to sing or, you know, to scream or anything else like that. Words are formed by the movement of our tongue, our lips, our cheeks, right? So all these different muscles that are in our mouth are helping us produce that sound. And then below the larynx is your trachea. Your trachea is your windpipe. It's a tough, flexible tube. It runs from that cricoid cartilage all the way down to where it branches to go into your lungs. And those branches to go into your lungs are called primary bronchi. The walls are supported by these tracheal cartilages, which you can even feel with your finger because your trachea is so close to the surface of your skin. You can feel the actual C-shaped cartilages that are there. And the cartilage is just there to make it more rigid, just to keep your airway open. That's why you have the cartilage there. If you look at a section, if you cut a section through somebody's throat and you're looking at it through a microscope, not that you need to look at it through a microscope, but if you're looking down, you'll see that the trachea is open and nice and wide. So here it is right here. This would be your trachea. This is one of the cartilages, and the esophagus is directly behind it. And the esophagus is more like a flat tube. There's not really any cartilage in there, right? It's behind your trachea, so it's protected in that way. One of the things that we don't often think about is, notice how your trachea only has cartilage kind of in the shape of a C. There's no cartilage at the back of it between your trachea and your esophagus. When you're swallowing food, your esophagus stretches, and so it can stretch into the trachea a little bit as well, right? This allows for that flexibility, so your esophagus can actually stretch this way and towards your spine as well, so that you can swallow, you know, larger pieces of food.
So when we choke, it's something that it's bigger than that can stretch.
Right.
Yeah.
So if you've pushed pushed
Okay.
the esophagus too far forward, then you're going to have a hard time getting air in.
That would be a big bite, yeah, for sure.
Yeah. So, and then the trachea ends up branching to go into the lungs. Those are called bronchi. You have two bronchi. You have a right and a left. The right one supplies the right lung. The left one supplies the left lung. The right one is at a steeper angle. Creates a more likely pathway for foreign objects. It's more likely that you could inhale something that would get to your right lung. And so here is a picture of the trachea. Here is your right bronchus, right? And here's your left bronchus. And then what you can see is what they're showing kind of in this light pink is the beginning of the lungs. So the main bronchi, which are also called the primary bronchi, only exist outside the lung. Okay, this is your trachea. This is where it branches to go to your lungs. Once it's in your lungs, it's already called either the lobar bronchi or the secondary bronchi. Those are interchangeable. Okay, and what that means is that this has already gone into your lungs, right? It's already gone some depth into your lungs. So the main bronchi are only outside the lungs. Once you get into the lungs, they're called secondary bronchi or lobar bronchi. And they're called lobar because each one supplies one of the lobes of the lungs, right? And so obviously we have two lungs. They are not equal. The left lung is smaller than the right. The left lung only has 2 lobes and the right lung has 3. Anybody have an idea why the left lung is smaller? What's there?
Because of the heart,
The
it
heart,
gives
right?
room to the heart.
You have a little notch cut out of your left lung, and your heart kind of sits to the left side of the center of your chest, and it kind of takes up some of that space of your lung. So you've got the primary bronchi, which are also called main bronchi. They divide into lobar bronchi. So now we're in the lungs. Those lobar bronchi divide into smaller branches called segmental bronchi. So they branch off the lobar bronchi, right? There are about 9 or 10 of these in each lung. And then these branches keep going, they keep getting smaller, and the smallest branches are called bronchioles, right? So you had primary bronchi, secondary bronchi, tertiary bronchi, which are what the segmental ones are, and then bronchioles. Bronchioles are not only smaller than the bronchi, but they are made of different tissue. So not only does the diameter decrease as you go down the lungs, right, but the percentage of cartilage decreases. In other words, there's a lot more cartilage in your trachea and in your primary bronchi. Your bronchioles have almost no cartilage in them. They're mostly smooth muscle, right? And the smooth muscle is involuntary, right? So smooth muscle can allow them to either constrict or dilate. So when somebody has asthma, for example, something will trigger their asthma. What it triggers is smooth muscle contraction. And so all these bronchioles constrict, right? And they make it really hard for you to get air in. And when somebody has asthma, oftentimes they have a rescue inhaler. They take a puff on that rescue inhaler, and it contains a muscle relaxer. And so what that does is it allows those airways to open up, right? And that's what allows them to start getting their breath back. That's what asthma does, and that's what those inhalers are doing. So this shows the bronchial branching. Again, your trachea comes down, branches into 2 main bronchi, which are also called primary bronchi, which branch into secondary bronchi, which are called lobar bronchi, which branch into tertiary bronchi, third, which are called segmental, which branch into these little tiny bronchioles. And you can see in this diagram, the light blue is the cartilage. And so you can see by the time you get to the bronchioles, there's really no cartilage. It's really all smooth muscle. And at the end, the bronchioles are called terminal bronchioles. At the end of the terminal bronchioles are the respiratory bronchioles. Respiratory bronchioles contain these air-like sacs called alveoli. Looks like a cluster of grapes, right? Each of those looks like a cluster of grapes. So we've got terminal bronchioles, right? And the terminal bronchioles branch further into respiratory bronchioles, and they lead to the alveolar ducts. So here we have a picture of your bronchiole, right? This would be a terminal bronchiole. This is a respiratory bronchiole. It ends in an air-like, or in a cluster of these air sacs called alveoli. So this looks like a cluster of grapes. Those are called alveoli, those clusters. What's happening is the air that we're breathing comes down through all these branches, and it ends up inside these little clusters. Now, surrounding all these little clusters, what do you notice? What do we have surrounding them?
Blood vessels.
Blood vessels.
Capillaries.
Capillaries, good. So smallest blood vessels, right? Capillaries, which are really small and have thin walls. It turns out that the alveolus, each grape, has a really thin wall around it as well. And that allows for the air, which is inside these grapes, to diffuse out across that wall and to diffuse into the blood, right? Capillaries have really thin walls. The alveolus has really thin walls. So air can diffuse out of these clusters and into the blood. This is where oxygen gets into our blood, is at these clusters, these alveoli. And all of this stuff that's going on is showing you the pulmonary arteries and the pulmonary veins. Remember, leaving the right ventricle of the heart are the pulmonary arteries, right? They don't have oxygenated blood. They have to go get oxygen. So they go to the lungs. They branch into smaller and smaller arteries and eventually branch into capillaries, which are surrounding the alveoli. Blue, right? They're still blue. They go from blue to red because they are getting oxygen from the alveoli. That's where you're picking up the oxygen. Now, these red vessels come back to a pulmonary vein. The pulmonary vein brings that red blood back to the left atrium of the heart, right? So it's all linked. That's why the respiratory system and the cardiovascular system are really closely linked together, right? So they're showing this as a branch of the pulmonary artery, which branches into an arteriole, which is really small, which branches into, capillary beds, right? And then from those capillary beds, you end up branching back into venules and back into the pulmonary vein. Questions about that? So the alveolar ducts and the alveoli are at the end of the respiratory bronchioles. They are the chambers that contain the gas that we're breathing in. Each lung contains about 150 million alveoli. Pretty much any organ in our body that we have two of, you can survive with just one. So you can survive on one lung and you can survive on one kidney. These allow for extensive rapid gas diffusion, which we need for metabolism. We need to make ATP, right? So again, you can see right here, here's the cluster. Each of these clusters are alveoli and they're surrounded by capillaries. What they're doing here is they're looking into each of those grapes, right? They've done the dissection and they've cut into that. So each of these cells is a simple squamous epithelial cell. What you can see are macrophages. So inside your alveoli, you have white blood cells that make sure that they're destroying any pathogens that are in there, because you don't want pathogens to get into your bloodstream, right? And so that's what some of these cells are. This purple one over here is an alveolar macrophage. The type 1 cells are just the cells that make up the actual alveolus, okay? And you can see that there are capillaries going around the outside of these alveoli. So you can see how thin that layer is. It's very easy for oxygen to get across that layer, or carbon dioxide to get back across the layer the other way. So I think we'll stop right there. We'll talk a little bit more about the lungs tomorrow. And then what I'm going to do tomorrow as well is go through some of that PDF that I posted about spirometry, about the respiratory system and what we're going to do for the respiratory physiology lab this week. So what kind of questions do you guys have for me?
I have a question. I was looking at the picture of the heart and I saw those two different sizes and lobes. Basing on their sizes, is the right lungs more effective than the left? Or
like do you exchange more gas through the right lung than the left?
right, because it's bigger, I don't know.
Yeah, that's a good question. I don't know if I don't know if it's enough to make a difference. Like I don't know if it's noticeably more. That I'm not sure of. You definitely have more tissue in your right lung, which would mean you have more alveoli, which would allow for more gas exchange. But I don't know if it's anything that can be, like, I don't know if it's big enough that it can be recognized, big enough difference.
Right. The only thing I found out is it's more vertical and they were more likely to get something stuck on the right lungs.
Right. The tube going from your trachea into your right lung is more vertical, and it is more likely that you can get, you know, particles in there. But I don't know that it's necessarily. I mean, there must be more gas that you're able to exchange, but I don't know that it's noticeably more or significantly more. That's a good question.
All right. Thank you.
You're welcome. Any other questions? All right. Well, then I will see you guys at 3 o'clock tomorrow, and we will finish up this chapter and we'll talk about respiratory physiology.
Thank you.
You're welcome.
Thanks.
Thank you.